Ready for a weekend? Yes. Should we just call it off here? What do you think? And then do twice as much on Monday? No. Nobody likes that idea. Okay, so last time I was rather hurried at the end, and so I was trying to do some beta-adrenergic receptor stuff, and I'm going to go over that in a little bit more care today. I'm not going to go into beta-adrenergic receptor in all the depth that we could, and that'll be a relief to you, because when we talk about glycogen metabolism at the very end of the term, that's where we actually go into that. So I'm going to show you some of the signaling aspects of it, and I'm going to show you how the signal gets turned off, but I'm not going to completely show you how the signal gets reversed, which is what happens. Okay? So, the beta-adrenergic receptor is interesting. It's a very, very important receptor because it allows the body to control things like glucose metabolism. And that's, as you know, a very critical thing because glucose is the primary energy source of cells. So, understanding how this signaling process works allows us to better understand how our body does what it does. So, as I showed you last time very briefly, the signaling process for the beta-adrenergic receptor goes through a series of steps like what I'm showing you on the screen here. Okay? And while I don't require you to memorize most of the signaling pathways, this one's pretty important. And we will use it later in the term. So I'm going to expect that you're going to know this signaling pathway that I show on the screen. The other ones I'm going to show you today are more complex, and I don't expect you're going to memorize them, but you should understand various features of them. So if we go through the steps in the beta-adrenergic signaling pathway, we see at the very bottom, of course, that what's happening is glycogen is getting broken down, forming glucose 1-phosphate. That's the G1P we talked about in this, or I sang in the song the last time. So let's just go through the steps in the process and see what happens. And what you're going to see in these signaling pathways that I'm going to show is this goes to this, goes to this, goes to this, goes to this. It's just a series of steps. Nothing overly complicated, okay? But they are a series of steps. So if we look at the first step in the signaling of the pathway is the hormone release and binding to the receptor. In the case of the beta-adrenergic receptor, there's really in the case of the glycogen signaling, which is occurring through the beta-adrenergic receptor, there's a couple ways in which this can get stimulated, and they come through different beta-adrenergic receptors. One of them will involve the hormone epinephrine, which is adrenaline that I talked about last time, and the other one can involve a protein called glucagon, and they both have pretty much the same effect. The primary difference being that glucagon is present in the liver but not the muscle, and epinephrine can work on all of these. Okay? So glucagon I like to think of as glucose all gone, right? Glucose all gone is telling the cell that makes glucose to start making some glucose, buddy, because we're running out of energy. Okay, second step in the process is that the receptor, which in this case is a membrane receptor, of course, changes shape on binding to the hormone, and on changing shape, it stimulates a G protein to load GTP and release GDP. Somebody asked me at the beginning of class, and it's a common question, is it a phosphorylation? Does that mean that we just put a phosphate onto GDP? And the answer is no. It's actually a replacement of the molecule. GDP is replaced by a new GTP. As I showed you last time, what happens with that is that coincides with the removal of the alpha subunit from the beta and gamma subunits of the overall 3-protein G protein. And now the alpha subunit is active, and it's going to participate in that signaling process. Prior to that, it's just sitting there on the beta-adrenergic receptor with the beta and the gamma doing nothing. Okay? Third step in the process, the alpha subunit with its GTP goes to adenylate cyclase and activates it. You can see that adenylate cyclase is also a membrane protein. It's a membrane enzyme. And what adenylate cyclase does, it catalyzes the conversion of ATP to make cyclic AMP. Cyclic AMP binds to protein kinase A. I'm going to show you that in just a second. And in the process of binding to protein kinase A, it activates it. Protein kinase A is, as I said, a kinase. You should always know when you see kinase that it's something that puts a phosphate onto something. So a protein kinase is something that puts a phosphate onto a protein. Yes? Oh, question, yeah. Okay. So in step 3, the alpha subunit, which is bound to GTP, goes to the adenylate cyclase and activates it. So adenylate cyclase is activated to convert ATP into cyclic AMP. Now, cyclic AMP is the second messenger here. We talk about second messengers. This is the second messenger. The first messenger was the hormone that bound to the receptor. This is the second messenger. As I said in class the last time, we tend not to talk about the proteins in the signaling pathway as messengers. We don't tend to call them that, even though that's what they're doing. We talk about the small molecules as being the messengers. So cyclic AMP is the messenger. It binds to protein kinase A and converts protein kinase A into the active form. We'll see how that happens in a minute. And protein kinase A is able to put phosphates onto other proteins. And you can see in this diagram that it puts phosphates onto 2 proteins. Okay? One is labeled GS, and one's labeled PK. The PK is phosphorylase kinase. The second is called Glycogen Synthase. Both of these proteins get phosphates put onto them by protein kinase A. And as you might imagine, we put a phosphate onto something, we change that something's activity, and that's exactly what happens here. I'll give you a second. Oh, question? I'm sorry. The what? So, good question. What's the green and the red? Red is inactive, and green is active. That's what I was actually getting ready to say. You read my mind. I don't know what you mean by knowing the actives. You have an inactive form and you have an active form. I'm sorry? No, they don't. They don't. In the case of the very last one, it does. Okay, but I'll talk about that in a second. Okay. So we've got the green form of protein kinase A. I'm sorry, phosphorylase kinase. You have the green form of phosphorylase kinase. You can see with the phosphate on, the green means it's active. And you can see the glycogen synthase has been converted from a green form to a red form, meaning it's inactive when it gains a phosphate. Oh my God, why so much confusion? Well, we'll see later that this is an example of what we call reciprocal regulation. You don't need to know that right now, but we'll talk about it later. Okay? Suffice it to say that glycogen synthase is getting turned off. Phosphorylase kinase is getting turned on. Was that a question? Oh. Okay. Now, it makes sense if you think about it to turn off glycogen synthase if we're going to be breaking down glycogen, right? We don't want to be making glycogen at the same time as we're breaking it down. Okay? All right. Well, the next step and the last step in the process is that phosphorylase kinase, which has been activated, now activates an enzyme that's labeled as GP. That's called glycogen phosphorylase. All right? So we see what I call a kinase cascade. Remember the cascade systems? This is a kinase cascade because we see kinase occurring at a couple of levels. Glycogen phosphorylase is the enzyme that breaks down glycogen. That's what's shown there. We see glycogen getting broken down into glucose 1-phosphate and a glycogen polymer that's shortened by one residue. Okay. So the result of the action of this hormone has been that this hormone has stimulated the release of glucose 1-phosphate. Glucose 1-phosphate, as we will see, can be used for energy very easily. That's what we call epinephrine adrenaline. Adrenaline causes, as I mentioned last time, release of a lot of glucose into the bloodstream. It gives a lot of energy for muscles, and that's needed if you're in a dangerous situation. So adrenaline has stimulated an increase in the blood glucose levels. That's the ultimate upshot of what's happening here. Okay. There's the 7th step. Questions on that pathway? Leo. Can I explain step 2? In step 2, what's happening is the hormone which has bound to the receptor has caused the receptor to change shape slightly, and that change in shape has affected the G protein that's bound to it. That change in shape causes the G protein to release its GDP, get its GTP, and release the beta and gamma subunits. So now that the alpha subunit is free to float around and do its thing. That help? Question? Yeah. So what's increasing with the binding of adrenaline is glucose 1-phosphate. Before this whole thing is active, there's no glucose 1-phosphate. But after this thing has been activated, glucose 1-phosphate is released from glycogen. Glycogen's already there. Okay? So all that this is doing is breaking down some glycogen that's already present. The glucose 1-phosphate can readily be made into glucose. Okay? The purpose of activating what? You're not activating glycogen synthase. You're inactivating glycogen synthase. We'll talk later when we talk about glycogen metabolism about activating it, and if we activate it, we would actually cause the synthesis of glycogen. That's what its name would suggest. So we want to turn that off if we're going to break down glycogen. That's actually what's happening here. Okay? Yes? It's a giant chain. It's a giant cascading system. One activates the next, activates the next, activates the next. Yep. Yes? What do beta and gamma do? Basically, they bind up alpha when it's bound to GDP and keep it from doing anything. Yeah. So that's and beta and gamma can actually interact with some other things in the cell that we're not going to talk about. But beta and gamma, for our purposes, that's basically what it does. Okay. Okay, so I promised I would show you how the protein kinase A is activated. So this was the enzyme in the pathway. That was actually enzyme at right there, number 4. So now we're going to see how that is actually activated by the cyclic Gmp. Okay, so if we look on this figure on the outline, you can see we are still up here at the top, okay? Yes? Not easily. So it's an issue that I'll have to work on, but this is a quick thrown together for today, so yeah. Okay, protein kinase A gets activated by cyclic Gmp, and well actually I can do one thing for you there, let me do that. So, Ahern. Let's make it. How's that? That help? Okay. Now you can. Oh, that's what he's talking about. Okay. Yeah. Sorry. Protein kinase A. I'll get back to that. Protein kinase A, folks, is activated by binding of cyclic Gmp, and this is how it happens. We can see protein kinase A has 4 protein subunits, 2 labeled C and 2 labeled R. The 2 labeled C are the catalytic subunits, and the 2 labeled R are regulatory subunits. It's similar to what we had when we talked about ATCase, for example. Binding of cyclic Gmp by the... why is it shown? Oh, okay. It's actually a little confusing how that's shown. I just realized that. Okay. It's not the catalytic subunits that's binding the cyclic Gmp. It's actually the regulatory subunits that are binding the cyclic Gmp. The little starburst there is to show that they've been released, but it's not the binding. The binding is actually occurring on the R subunits. Okay? The R subunits bind cyclic Gmp, and when that happens, they release the catalytics. The catalytics can go out and do their things, which means they are now able to put phosphates onto proteins. And that's what they do. In the absence of cyclic GMP, the regulatory subunits will bind to the catalytic subunits, and there will be no release, and there will be no catalysis. This is a good example of a perfect on-off switch, because the regulatory subunits cover up the active site and prevent it from being able to do anything. So when we said allosterism, we had a volume up and volume down because we didn't turn it all the way on or turn it all the way off. Here, it's an on or off because we're either having the catalytic subunits open or not open. Yes? Say it again. What happens when it binds? Okay. So we've got protein kinase A in the top form. The regulatory subunits are covering up the catalytic subunits. And when they cover up the catalytic subunits, the active site is not exposed. No activity from this enzyme. It will not phosphorylate anything. Okay? Binding of cyclic GMP by the regulatory subunits causes them to let go of the catalytic subunits, and now the active site is open. At that point, the active site can catalyze the phosphorylation of other proteins. That's what protein kinase A does. And as long as those catalytic subunits are free, that's what's going to happen. Is that clear? Does that help? The cyclic AMP is binding to the regulatory subunits. The starburst is not the binding site. It's just indicating it's been released. But the cyclic AMP binds to the regulatory subunits, the Rs. Okay. Well, I've shown you how to activate the pathway. You activate the pathway by having a hormone bind to the receptor. We talked about in class earlier in the term how enzymes are dangerous because enzymes are very active. And enzymes can break down things very rapidly, which is good, but if they break down too much, that's not good. If we let this enzyme just go, the cell would break down all of its glycogen, it would lose all of its energy, and it would die. So being able to turn off this enzyme and turn off this pathway is as important as being able to turn it on. Now, there are several things that are involved in turning off this pathway. I'm going to show you 4 of them. I'm not going to show you all of them because we'll actually talk about the rest of them when I talk about glycogen metabolism. But let's talk about the 4 here. The receptor, pretty critical in the process. If the receptor, for example, doesn't let go of the epinephrine, what's going to happen? It's going to continue signaling, it's going to continue favoring the activation of the alpha subunit, and that is going to continue favoring all the rest of the things here. There are times when the receptor gets stuck. And so I'm going to show you what happens if the receptor gets stuck. Okay? So that's one of the things we have to think about in turning off the signal. The second thing we have to think about in turning off the signal is the G protein. Because the G protein, if it gets stuck, it's going to leave this cell in a scheme that's going to continually be breaking down glycogen. We don't want that. The third thing, cyclic AMP. If the cell doesn't get rid of cyclic AMP, what's going to happen? Well, it's going to also continue doing that pathway all the way through. The last thing is protein kinase A. If protein kinase A is left on, then protein kinase A will continue the phosphorylation and we will continue the breakdown of glycogen. So all 4 of these things are critical to making sure the cell is breaking down glycogen when it needs to and not breaking down glycogen when it doesn't need to. Let's talk first about the receptor. It's fairly simple. Here's the receptor. We think the exterior is at the top, interior is at the bottom. It's at the bottom where the alpha subunit and the rest of the G protein would interact. If the receptor gets stuck, okay, what happens is there's an enzyme called the G protein receptor kinase that will put a phosphate onto the bottom of that receptor on the cytoplasmic side. When that phosphate gets put onto there, there's a protein called arrestin that will bind to it, cover it up, and prevent the G protein from interacting with it. We've stopped the signaling from the receptor end. Now, this primarily happens when the receptor is stuck. Okay? And it does happen. Okay? Endocytosis means it gets internalized. There's probably a reason why that thing is stuck. Let's get it in. Let's not leave it out there to continue signaling, and the signal goes away. G protein. The G protein has to be inactivated because if it's not inactivated, as I said, we'll continue the signaling process. This is a cool aspect of the overall signaling. The G protein actually controls itself. And it controls itself because it is an inefficient enzyme. I'll repeat that. The G protein, the alpha subunit of the G protein, is an inefficient enzyme. So far, you've just seen it signaling. You haven't seen it catalyzing any reactions. Now I'm going to show you the reaction that it inefficiently catalyzes. Okay? It has the ability to cleave GTP. Break down GTP. It's carrying GTP. It carries GTP, and if it were an efficient enzyme, it would immediately break it down. And the signal would be turned off, and nothing would happen. There would be no breakdown of glycogen. But it's an inefficient enzyme, meaning it doesn't work very fast. As long as it's carrying GTP, the protein is active. And when it's active, the signaling continues. And as over time, it breaks down the GTP, it becomes inactive. So it's turning itself off. Schematically, it looks like this. Here's the alpha subunit bound to GTP. Hydrolysis leaves us GDP. And when that happens, the beta and the gamma subunits can then bind back to it again. And it can go back to another receptor and bind to it. Question? The question was, if I understood it right, was is this an on-off switch? GTP versus GDP, the answer is yes. Okay? So if that's what you mean by on-off, I would say yes, because in the GDP-bound form, it will not activate adenylate cyclase. And in the GDP-bound form, the beta and the gamma come back, and it gets hauled back to another receptor. Yes?
So essentially,
it burns through its GTP. It's only bound to one. So there's only one it's bound to. But it has to break that one down before it becomes inactive. If it can't break that one down, and we'll talk probably not today, but on Monday, if it can't break that down, the cell's got some problems. And we'll see some of those. Okay? Okay. Returns to the resting state, waiting for a GTP. So at this point, we have inactivated the adenylate cyclase because we have not stimulated it with the alpha subunit. The alpha subunit is gone. It's no longer there. Okay, the third and fourth parts of the cycle are here. Okay, so we've seen that we've blocked that. We've seen that that can't happen, and in the consequence of that, we've also knocked out adenylate cyclase. In this step of the process, we're going to break down cyclic AMP. Yes? Question? Yes?
So you mentioned the beta and gamma subunits moving back to the cell.
Yes.
Aren't they attached to the cell wall? Do they do a whole lot of movement?
So all this is happening around the cell wall, but there's mobility. It's not, the cell wall is fluid, okay? And it's not cell wall, it's a cell membrane, but it's fluid. So it's not like that things can't move around, no. Yeah, okay? Okay. Now, I said cyclic AMP was the third thing we had to think about. Cyclic AMP gets broken down by an enzyme called phosphodiesterase. And when that happens, it gets converted into AMP. Well, if you have no cyclic AMP, you're not going to have any active protein kinase. And you don't have any active protein kinase, you've essentially shut the whole signal down. All right? So we've now shut off the four things that were really stimulating that cycle to occur, or that signaling pathway to occur. Okay? I'll show you something cool. Here's the cool. The cool is that phosphodiesterase is inhibited by caffeine. What does that mean about you getting a buzz for your coffee? Well, I'll tell you what it means. If the phosphodiesterase is inactive, it means that cyclic Gmp is not going to be broken down. And if cyclic Gmp is not going to be broken down, that signaling pathway to release glucose into your bloodstream is going to continue to run. That's why one of the reasons you get a buzz from your coffee. You up your blood glucose. Caffeine is doing that. That makes sense? Everybody's smiling now. They must have been the coffee drinkers, I can tell them. Oh, this is good, right? Now I know. All right. So, it gives a boost to blood glucose. Okay. Okay, so I've now gone through the inactivation. You've seen the coffee buzz. I want to turn our attention. Questions, may I say? No? Okay. Turn our attention to another kind of receptors, another set of receptors. So the beta-adrenergics are one set. All right? Another set are the receptor tyrosine kinases, or RTKs. Yes? 4 steps, yes. The last one was pKa because there's no cyclic Gmp. So when there's no cyclic Gmp, pKa will be inactive. Yeah. Receptor tyrosine kinases, RTKs. So these are also membrane-bound proteins. Okay? These are membrane-bound proteins. And what they do is they phosphorylate tyrosine residues in other proteins. Okay? So kinase, of course, putting phosphates onto things. They're membrane-bound proteins that phosphorylate tyrosines in other proteins. And it turns out that phosphorylation of tyrosine is a relatively rare thing, and it plays very important roles in decisions of cells to divide. We'll see how that plays out in a couple of these. Play important roles regulating cell proliferation. Okay? One of the important things about most of the RTKs, not all, but most of the RTKs, excuse me, is that they must dimerize to be active. And it's the binding of the hormone that causes them to dimerize, as I will show you. Before I show you that, I will show you the activity that they catalyze. It's simply a phosphorylation of tyrosine. You see the tyrosine side chain on the bottom. You see a phosphate coming from ATP, and you have a phosphorylated tyrosine. That phosphotyrosine affects the activity of the protein that contains it. And so this is a way of controlling what that protein does. Here, in the cell membrane, are 2 receptor tyrosine kinases. As they're shown, they're inactive. They're not bound to hormone, and they're not dimerized. There's the lipid bilayer, inside on the bottom, outside on the top, and there's the monomers. Okay? Binding of the hormone causes these guys to dimerize, so they come together. Okay? They're coming together. The coming together of those causes them to do something to each other. They do something called autophosphorylate. And what that means is that on the inside, that is the cell side, the bottom, phosphates get attached to each of them. So one does the other, back and forth. You can see several phosphates get put on, and they get put onto tyrosines. And that causes this dimer to become active. So we just activated this dimer by this autophosphorylation that happened because dimerization occurred. And dimerization occurred because the hormone bound. Okay, what happens next? Those phosphotyrosines are targets. They're targets for binding by cellular proteins. This is an important thing. They're targets for binding by cellular proteins that have a specific binding domain that recognizes phosphotyrosine. That domain is called an SH2 domain, as we shall see. So I've schematically shown some proteins. I've schematically shown the signaling complex that's coming in. And we can see in this case that, let's say, the green guy that's there has an SH2 domain that's recognizing and binding to one of those phosphotyrosines. Okay? Now, there's a lot of steps. I'm just going to show them to you. I'm not going to expect you to memorize them. There's a lot of steps. But the result of this is that the signal is now on the inside of the cell. Okay? The signal transduction has occurred. The signal is now on the inside of the cell. Let's schematically look at what's happened. Bound the hormone. Dimerized the receptor. Autophosphorylated the tails. Assembled the signaling complex. Communicated the message into the cell. Now I'm going to take you from that point now and show you some steps in the next part of the process. I'm going to talk about the insulin receptor. The insulin receptor is a slight exception to what I've already told you. The slight exception is it doesn't require insulin to dimerize. It's already a dimer. And you can see it's a dimer on the screen. Most RTKs don't do this, but insulin does. Insulin receptor does. And there's a difference between insulin and the insulin receptor. Insulin is the hormone. Insulin receptor is the thing that's bound in the membrane. Okay. Always a dimer. It does, however, take the binding of insulin for the autophosphorylation to occur. Autophosphorylation will not occur unless insulin binds. Binding. Autophosphorylation. Now I'm going to go through a bunch of steps, and you can just sit back and relax and watch them. How's that? It's a protein called IRS1 that gets activated. IRS1 can either go to other signaling pathways, or it can activate the next protein in the signaling pathway here, which is a kinase. We see a kinase cascade, which favors formation of a molecule called PIP3, which activates another kinase, which activates another kinase, which now does something that you should know. So this kinase cascade has resulted in a targeting of a protein called GLUT4, G-L-U-T-4. And that protein is moved from being inside of the cell to being moved to the membrane of the cell. Why is that important? Well, here's why that's important. GLUT4 stands for glucose transport. The action of insulin has stimulated the cell to move a glucose transport protein to the cell membrane. And when it's on the cell membrane, what's it going to do? Transport glucose, right? It's going to move glucose in. That's what insulin does, folks. Your body makes insulin when your blood sugar goes high. And as I talk about in this class, and I'll be talking about the rest of the term, glucose is a poison as far as your body is concerned. I'll repeat that. Glucose is a poison. High levels of glucose will kill you, can kill you, damage your cells. People with diabetes have high blood glucose. Glucose is important, but too much of a good thing is bad. Glucose is a poison. If you have high blood glucose, the body releases insulin, and it stimulates cells to do exactly what you see on the screen here. Take in glucose. But glucose is a poison, Ahern. Right? Glucose is a poison. Inside the cell makes it into glycogen, where it's no longer a poison. Okay? So, high blood glucose is fought by insulin by bringing the glucose into the cell. And this is how it does it. Cells uptake glucose, blood glucose levels fall, And we'll see that last point, which won't have too much significance to you at this point. But it stimulates action of a protein called phosphoprotein phosphatase. Phosphatases do exactly the opposite of kinases. They take phosphates off of things. You start to say, oh, this is going to do something opposite to epinephrine. Yes?
So is anti-diabetes have to do with that receptor, or does it have to do with the pathway?
Does type 2 diabetes have to do with the receptor, or does it have to do with the cascade? Type 2 diabetes comes in many forms, but it tends to most commonly result in what's called insulin sensitivity. Cells aren't responding to it, and that would be receptor mediated. Okay? But there's many types of type 2 diabetes. All right, now I've shown two pathways to, yes? What makes it a poison at high levels? It gives osmotic shock to cells. Osmotic shock to cells. Too high of a level, you got a problem. You start bursting cells. Yep. Okay. Here's two pathways I'm going to show you work exactly the opposite of each other. The beta-adrenergic we saw was stimulating the release of glucose, and we're going to see that the insulin pathway is going to favor the use of glucose to make glycogen. There's the beta-adrenergic pathway. The upshot there is that glycogen is broken down, and blood glucose levels rise. Insulin, on the other hand, through all these things that I've shown you, and by the way, you don't need to know the steps in that insulin pathway. Okay? So don't worry about the steps. The upshot is that glucose is taken into the cell. We also saw that phosphoprotein phosphatase was activated. It's going to take the phosphates off of all those proteins that protein kinase A and phosphorylase kinase put phosphates onto. Consequently, we're going to reverse the beta-adrenergic pathway. Those are exactly opposite of each other. One favoring blood glucose levels, one reducing blood glucose levels. And I hope that you can tell me why that happens. Okay? Questions on that? Yeah? Oh, okay. So good question. So very good question, actually. Her question is the insulin pathway exactly the same as the inactivating pathway that I showed before? All right. The effect is the same. You stop the signaling. But it's even more than that in that in this case you're going to stimulate, as we will see later, the synthesis of glycogen. You actually will stimulate the synthesis of glycogen. But that's a little further down the line. But very good question. Okay. Let's see. How much time do I have? We'll get started on this. All right. So I've talked about the insulin receptor. Let us think about the epidermal growth factor receptor. The downside, if I make it too big, then I can't fit it on the screen. Let's see, Ahern. Hopefully that'll stay. Okay. So we saw now how the body's responding to glucose. And this turns out to be really critical. The body has a very narrow range in which glucose levels are good, if we want to think about good and bad. Very narrow, acceptable range. Too much glucose, poison. Too little glucose, you have hypoglycemia. Okay? People who have insulin not uncommonly develop hypoglycemia, and the reason they develop hypoglycemia is that their insulin response is not in sync with the glucose levels. Right? So let's imagine that I eat a bunch of glucose. Glucose levels rise. If everything's working okay, insulin levels rise, and when that happens, it starts pulling down the blood glucose level, right? However, if the body isn't responding, let's say we're the type 2 diabetic that is not having their cells respond properly to glucose, what happens? Blood glucose levels rise, insulin starts rising, blood glucose levels keep rising, body starts making even more insulin, And all of a sudden, things start to kick in, and blood glucose levels start falling, and insulin's way up here. If insulin levels are high and blood glucose levels are falling, what's going to happen to your blood glucose levels? They are going to crash. And that's how you become hypoglycemic if you have diabetes. So people who have diabetes will have large swing, or can have large swings in their blood glucose levels. High when there's very little response, and then low when the body over-responds with insulin, and all of a sudden everybody wakes up and says, oh, I've got to take in all this glucose. And there's no glucose to take in. Our brain and our eyes are very, very dependent upon the levels of glucose in our body, because our brain and our eyes cannot make glucose, and they're two of the biggest users of glucose. So they're dependent upon our liver, primarily, to modulate and provide the levels of glucose that are necessary for them to function properly. That's why this system's important. Okay. I'm just going to introduce this, and then we'll go to the song. Epidermal growth factor is another RTK. Okay? Back up. Epidermal growth factor receptor is another RTK. That should be changed, shouldn't it? Now I'll do that later. All right. Epidermal growth factor receptor, ETK. Okay? Epidermal growth factor, EGF. The epidermal growth factor receptor, EGFR, is an RTK. EGFR is an RTK. Okay? You see the receptor on the screen. You see it plays very important roles in growth of cells, proliferation, and cell differentiation. Right? There's the EGFR, that's the receptor. There's the binding, there's the EGF, the epidermal growth factor that it binds. So the epidermal growth factor receptor binds the epidermal growth factor. They're both proteins. They're both proteins. Okay? Here's what the signaling looks like. Now, in the case of the epidermal growth factor receptor, it's a monomer and becomes a dimer when it binds. It's not like insulin. Insulin started out as a dimer. Epidermal growth factor receptor starts out as a monomer, comes together, and epidermal growth factor receptor also autophosphorylates. It also is a tyrosine kinase. It also has the assembly of a signaling complex. You can see the signaling complex on the screen. Now, there's one protein on the screen I'm going to talk about a lot next time. It's called RAS. Question? Yeah? Do all RTKs autophosphorylate? The answer is yes. They do. Yep. RAS is an example of what we call a proto-oncogene. And oncogenes, when they're proto-oncogenes, when they're mutated, become oncogenes, and those are involved in cancer. Now, I told you that the consequences of signaling being screwed up can be very problematic, and we're going to see that next time. But before next time, we are going to move to that doesn't sound good. I am missing meals on busy days. That's when my body steals glucose away from my liver. It starts with glucagon when I'm weak-kneed. The hormone acts to spawn new energy in my liver. The signaling acts rapidly. CAMPs fire up kinase. Phosphorylase then gets reactivated. So glycogen begets glucose phosphated in my liver. Step here of phosphatase makes phosphate disappear with no delays in my liver. Okay, folks, see you on